So today we're going to talk about mutations and genetic variation, and then get into some biotechnologies that are used today to produce products on a mass scale. So just by general definition, a mutation is any change in the sequence of a DNA molecule, and therefore potentially produces a different protein or an incorrect protein. Now, sometimes these mutations are random, and sometimes they're acted upon by natural selection, but in any case, when mutations occur in a cell on a DNA level, every time the cell replicates, whether it's through mitosis for body cells or meiosis for gametes, eggs and sperm, that mutation is going to be repeated and repeated and repeated in every cell after that. A beneficial mutation, like we talked about in Biology 20, if it's beneficial, that means that it has some type of selective advantage for the organism, potentially giving it more opportunity to survive. If it's harmful, then that's going to make it less likely to survive, according to Darwinian theory. And sometimes mutations can be neutral. So this means that they are neither helpful nor harmful to the organism in terms of its ability to survive long term. So when we talk about a gene mutation, that's the term that's given to any change in that sequence of base pairings. Okay, so maybe you have a T where a C should be, or a G where an A should be, and that can ultimately affect how proteins are ultimately produced. So when we talk about mutations, there's several different kinds that we can discuss on a gene level. So the first one is a point mutation, and a point mutation, as the name implies, is something that affects just a single base pair in a DNA sequence. They may or may not cause a change in the amino acid coded for by that codon. Remember from your mRNA codon chart, we have that duplication of the triplets that can make the same amino acid. So sometimes the mutation will code for the same amino acid, and therefore the organism in the cell is not impacted. But they only have the potential to affect one amino acid in the protein. So quite often these are not super severe, but they can be of three particular types. They can be silent, and as the name implies, if the mutation is silent, that means that the particular mutation in the base pair doesn't actually alter the protein, because that same nucleotide actually produces the same amino acid. Okay, so if it's silent, it doesn't change what's produced. If it's a missense, kind of just think about what that term means. If it's a missense, then that means that there is a change in the amino acid, and ultimately the protein product can be altered at the end of transcription translation. And then you also have something called a nonsense mutation. And remember, if something is nonsense, it means that it can't even happen, it doesn't even make sense. And what happens then is sometimes you'll get a change in a codon, and what it does is it actually makes a stop codon. So you have the DNA strand, you know, the transcription and the translation happening, and then all of a sudden you get a stop codon where it shouldn't be, and then the ribosome detaches and it finishes before the protein product is even done. And as a result, that can very severely impact protein production, and quite often among the three types of point mutations, that one is the most catastrophic, or has the potential to be the most catastrophic. So if we look at this on a kind of visual point of view, we can kind of see what we mean here. So when we have no mutation, so this is kind of our original production of that amino acid, okay? So on the DNA level, we have our triple T, and then on the mRNA level, we have our triple A, and that produces lysine. And so the key here is it's silent, because if you look at your mRNA codon chart, AAG produces lysine, but so does AAA. Okay, so there's no change in the amino acid actually produced. Now in the next one, which is the nonsense mutation, okay, remember this one is going to produce a stop codon where one shouldn't be. So if the mutation produces UAG instead of AAG, okay, see how we have the U here, then that actually is a stop codon, and so that ribosome is going to release that mRNA strand, and we're done. Okay, we're not producing any type of complete sequence of amino acids to make a protein. And then what we have is, our last one is that missense mutation, and with the missense mutation, notice that we're producing a different amino acid. So we have AGG, which produces arginine instead of lysine. Okay, so that is going to affect the amino acid production. So if we look at the visual here, here's the amino acid that we want, right? Silent produces the same amino acid. Stop codon produces no amino acid, and then missense produces the wrong amino acid. Okay, so those are kind of our three main categories of gene mutations. So what can cause mutations? There's a number of things that can cause mutations, but more often than not, they are spontaneous. And what that means is when DNA is replicating, there's errors that are made. So remember, we talked about our DNA polymerase being like our builder, but also our proofreader. And so the DNA polymerase goes through and it checks to make sure that all of the base pairs are matched up properly. DNA polymerase isn't perfect, and especially as people age or organisms age and the DNA replication just isn't as good as it used to be, spontaneous mutations can happen. Okay, so there are errors in replication that are just not caught in that DNA replication process. So a lot of genetic mutations are very simply spontaneous. But we can, of course, be influenced by our environment. So induced mutations are mutations that result because of exposure to what we call mutagens. And a mutagen is anything that can cause a mutation in DNA. So that includes things like ultraviolet radiation, includes like things like X-rays, chemicals, etc. So those can all cause DNA replication to be incorrect because of that environmental impact. Now, a carcinogen is a specific term that we use for agents that actually cause cancer, okay, cancer-causing agents. So these are a very specific type of mutagen that will cause DNA replication to become erroneous to the point where the cell can't actually do what the cell is supposed to do anymore. Okay, so it's like this rogue cell, and it will multiply really quickly, accumulating all of these errors, getting out of control, and sometimes overtaking the healthy cells. So we all have cancer cells all the time in our body. It's just that usually our immune system takes care of them before they are able to get out of control. So remember, though, that mutations can not only occur in that nuclear DNA, they can also occur in mitochondrial DNA. So keep in mind that there's two different types of DNA. Nuclear DNA in eukaryotic organisms is that DNA that's housed in the nucleus. It's that super long strand, and it's very large. And so sometimes when scientists are doing research on DNA, they will use the DNA that we find in mitochondria and in plant cells' chloroplasts. Okay, so mitochondrial DNA is found in all organisms that have membrane-bound mitochondria, and then organisms like plants that have chloroplasts, we have specific types of DNA in there as well. And so because these DNA strands in the mitochondria and the chloroplast are smaller, they actually are much easier to study and much more efficient to use for purposes of like tracing lineage, for example, of human beings. So mitochondrial DNA is abbreviated mtDNA, okay, and this is in both the program of studies for Alberta as well as IB. And mitochondrial DNA is used to trace inheritance through the maternal line only. Now, if you remember so in other words, offspring don't get mitochondrial DNA from the biological father, they get mitochondrial DNA from the biological mother. And if you remember before, we kind of talked about, when we talked about monozygotic and dizygotic twins, we talked about the fact that the sperm cells are actually super, super small compared to the egg cell. And so really what the sperm cells bring to conception is they bring chromosomes. They bring the 23 chromosomes from the father. But the egg cell, because it's so much larger, the egg cell is what has the cytoplasm, the enzymes, and the organelles that are necessary for cell division, and that includes the mitochondria. So the reason why mitochondrial DNA always has to be traced through the maternal line is because we don't actually inherit any mitochondrial DNA from the father. Okay, the egg is the only source of the mitochondria passed on to the offspring. So what's the whole point of mitochondrial DNA studies? Basically, what is done is because it's a very small chunk of DNA, and because we know that it is from the maternal line only, scientists can actually take that DNA and then trace it back through human origins, for example, for the out of Africa hypothesis, as well as using it to prove relationships with deceased individuals, like with the last imperial family of Russia, who were actually identified once their remains were found as conclusively being that family based on the mitochondrial DNA studies compared to living relatives. Okay. So now we're going to transition to DNA and biotechnology. And one really, really important term here is recombinant DNA. And I want you to think about what that word recombinant means. It means to recombine DNA. So really what we're doing here is we're creating a new type of DNA to actually produce a product that is desirable to make on mass production in the laboratory. Okay, so what's actually done with recombinant DNA is the gene that codes for a certain protein like insulin, like human growth hormone. Okay, we know where that gene is because we know what our human genome is now, right? So what they do is they find that gene, they isolate it, they cut it out, and then they put it into what's called a plasmid, which is a little circular type of piece of DNA found in bacterial cells. And then what will happen is that little bacterial cell plasmid will actually start to produce lots of insulin or start to produce lots of growth hormone or whatever it has been recombined to make. Okay, so back in the day, scientists, when, for example, insulin was discovered as being a treatment for diabetes, that hormone had to be harvested from the pancreas of animals like pigs or cows or whatever. But now recombinant DNA is used to produce insulin for diabetics, and it's much more efficient, it's cruelty-free, and it's much more cost-effective as well. So basically, when we introduce foreign DNA into a living organism, it's called genetic transformation. Okay, so that's just a term. And the whole idea with this type of biotechnology is to trick an unsuspecting bacterial plasmid, little ring of DNA, into making a protein that it typically does not make. Okay, so you're actually giving it the gene necessary to produce a protein that it would not make in its natural world. Okay, and this has led to, of course, a lot of advances, as we said, in biology, agriculture, and medical technology. So let's talk a little bit about enzymes and recombinant DNA. So enzymes, remember, are catalysts, right? So what enzymes do is they facilitate some type of metabolic reaction more efficiently and quickly without too much heat or too much energy on the part of the cell involved, okay? So what scientists do is they actually use these very special enzymes called restriction enzymes. Sometimes they're called endonucleases, okay? But for our program of studies, they're called restriction enzymes. And what they do, first of all, is they identify what gene they actually want. Okay, so what gene are we looking for? Do we want the gene that makes insulin? Do we want the gene that makes growth hormone? What are we actually looking for in terms of that production of that protein? So they identify where that gene is on the donor cell, okay, on the DNA of the donor cell. And then what they do is they introduce specific restriction enzymes, okay, to that segment of DNA where that gene is. And what the enzymes do is they actually cut, they're like little scissors, and they actually cut out that special gene that is desirable on the part of the scientists. And then what they do is they take that gene that's been cut out and they insert it into a recipient cell, recipient DNA ring, plasmid, and then what we have is recombinant DNA. So looking at that in a little more detail, restriction enzymes, as we said, sometimes called restriction endonucleases, are like scissors. And what they do is they cut a double-stranded DNA at a specific base pair sequence, and this is called the recognition site. So when this enzyme is introduced in a laboratory, in a petri dish, okay, when that enzyme is introduced into the DNA strand in question, it literally has, based on its active site, remember we talked about active sites in grade 11, so based on its active site and what it can actually attach to or work on, it actually finds that recognition site in the gene and it actually cuts the gene out, okay, in preparation for inserting that gene into the receiving bacterial cell plasmid. Most recognition sites are either 4 to 8 base pairs long, so they're not huge, and they are palindromic. And remember what palindromic means? It means they're the same forwards and backwards, okay? So if you look at the word mom, right, that's a palindrome. So if you spell it one way or in the reverse, you still get the word mom, right? So many restriction enzymes leave these little, they're called sticky ends, or they're like little overhangs where they actually do the cutting. So if we take a look at what that means here, these are just examples of different restriction enzymes. Now in our program of studies, you don't have to memorize any of these restriction enzymes. It'll always tell you, you need to know what a restriction enzyme is, but they'll always tell you that restriction enzyme EcoR1 is introduced into a DNA strand, blah, blah, blah. Okay, so you don't have to memorize any of these particular restriction enzymes. So what we mean by sticky ends is this. So first of all, you see where these little red arrows are here? Those little red arrows are the recognition sites. And so when that enzyme is introduced to that DNA strand, what it actually does is it cuts out the particular sequence of codons or sequence of base pairs in question, and then it leaves sticky ends. So any of these that you see that have this little overhang, see how the G is like a little overhang here? That's called a sticky end. Now you'll notice with the next restriction enzyme here, it doesn't produce sticky ends, right? So these are called blunt ends. So see how they don't have those little overhangs, okay? They're blunt ends instead, okay? So that's just a different type of enzyme, cuts in a different way. So some restriction enzymes produce these sticky ends where they have that one codon, or sorry, that one base pair overhanging, and some of them have the blunt ends, okay? But the whole point here is after the restriction enzyme, we call it digestion, after the restriction enzyme digests the DNA strand and cuts out those base pairs in question, then we end up with either a sticky end situation or a blunt end situation, okay? Now in terms of the palindrome piece, this is what we're talking about. So see, for this one, this blunt end here, we have GGG, CCC, and then we have CCC, GGG, okay? So they're opposite from front to back. So restriction enzymes are very precise and very predictable in terms of how they actually will cut out that gene of interest, okay? And if they cut the DNA of the organism that they want to insert with the same restriction enzyme, the foreign DNA has the same sticky ends, okay? So what does that mean? So you have a restriction enzyme introduced into the original DNA strand, and then you have a restriction enzyme introduced into that circular plasmid protein, which is going to receive, or plasmid DNA rather, which is going to receive that gene. So you put the restriction enzyme here and it cuts it out, and then you put the restriction enzyme here and it also cuts it out, leaving those overhangs so that this piece of cut out DNA can be inserted directly where the other piece was cut out. So let's just take a look at our graphic here so we actually can see what we're talking about. So here we have our original DNA sequence up top here, right? And these little dotted lines right here, that represents the recognition site. And the red box area here, that represents the gene of interest, okay? So whatever the gene is, the insulin producing gene, the growth hormone producing gene, whatever. So basically in this case, the restriction enzyme is EcoR1 again. We know that's one that leaves those sticky ends or those overhangs. The restriction enzyme is introduced. It goes to those recognition sites and it digests or cuts out that gene of interest, okay? And then the end result is this situation here, where here's the gene of interest, right? That red box, just like from above, which has the code we want for a specific protein. And then you have these sticky ends, these overhangs. And remember, the overhangs are not to produce whatever hormone or protein or whatever we want. The sticky ends are so that this cut out piece will then attach itself, because of the desire to match the base pairs, to the sticky ends of the receiving DNA. So once the gene of interest has been removed from the DNA molecule, it can be inserted into that new piece of DNA, which is now called recombinant DNA. Okay, so once the receiving little plasmid, little circle of DNA gets that new gene, it's now called recombinant DNA because it's a combination of its own genetic material plus that gene that was cut out by the restriction enzymes. So what enzyme have we studied that glues DNA into place? That is ligase. And remember, ligase is still used here, so it's added to actually fuse together and glue together the inserted gene into that receiving DNA plasmid. The complementary sticky ends match up because remember, you always want our A's and our T's together, or sorry, our C's and our T's together, our C's and our G's together, and so those will be attracted to one another and those sticky ends will connect. And then again, the ligase is added to make it all fuse and be actually productive. So here's another kind of good diagram. I quite like this one here. So what we have here is we have a plasmid. Now remember that a plasmid, we talked about this before and we'll look at it in another slide too, but remember that a plasmid is a very small little circle of genetic material that is found in bacterial cells. So bacterial cells will have that long DNA strand, okay, that looks like that just piece of yarn all in the cytoplasm, but then they also have these little circles of DNA called plasmid DNA, and these are very desirable for recombination because they're small, and so they're really easy to work with, and it's really easy for the restriction enzymes and the ligase, etc., to do what they need to do. So, okay, here we have our chromosomal DNA, and this is going to be our donor DNA, okay? So we have found the site that we want, okay, and the cleavage site just means where it's going to cut, right? So we have the recognition sequences, so the restriction enzyme knows where to go. And then when the restriction enzyme is added, so this time we're actually adding two, so we have the ECO R1 and we also have the PUV L, the PIVO. So essentially we add the restriction enzymes to the donor DNA. The restriction enzymes cut out the gene of interest, and notice that the first restriction enzyme gives us that sticky end, and the second restriction enzyme gives us our blunt ends. Okay, so again, it just depends on the enzyme. But in any case, they have both cut out that gene of interest because that gene of interest is what's going to produce the protein that is the end desired result, okay? And then what they do is they add the same restriction enzymes to our little plasmid. So then we end up getting a sticky end, right, for our first cutout piece of DNA to attach to, and we end up getting a blunt end for our second piece of desired DNA to bind to. So the same restriction enzymes are added to the plasmid as to the original chromosomal DNA so that we get an attachment site, okay? So then what's done is those two pieces of DNA, so the one with the sticky end and the one with the blunt end, so you can see the purple here, right? Attach at the appropriate site, cut out by the same restriction enzyme, and the one with the blunt end also attaches to the new plasmid, and then ligase is added to bind everything together nicely, and now we have recombinant DNA. So this yellow piece is the original DNA from the plasmid, and then the colored regions here, that's the recombinant part. So that's the genes of interest, so two in this case, that were taken from that original donor chromosomal DNA. And then the whole thing together is called recombinant DNA because it consists of some original, but it's been recombined with new genetic material to produce a new product. So essentially, by cutting out part of the plasmid DNA and inserting the genes of interest from the donor chromosomal DNA, we actually have now tricked the plasmid DNA into producing the protein of interest, okay, whatever that protein is. So then what will happen is these plasmids, once they've been engineered and they are now recombinant DNA, they're going to be inserted back into the bacterium. And then guess what? They're going to start producing insulin or human growth hormone or whatever particular protein is desirable in terms of production. So transformation is just a general umbrella term for any process by which foreign recombinant DNA is incorporated into the genome of a cell. The vector is a term given to the vehicle, and that's IB. Okay, this is not Alberta Program of Studies. This little term is an IB term. The vector is a vehicle by which foreign DNA may be introduced into a cell. And as we said, the goal of most genetic transformation is to change the traits of an organism or change what that organism actually produces on a cellular level. Okay, anytime an organism has foreign DNA in its genome, it's said to be transgenic. Now, this is not cloning. Okay, cloning, offspring is identical to parent. Okay, so that's not a change in DNA. A change in DNA is when you actually genetically modify an organism by removing part of its genetic sequence and replacing it with something else. Okay, so just kind of keep that in mind. They're not exactly the same thing. So as we talked about, bacteria is used quite often to make a useful gene product, such as we said human insulin, human growth hormone, etc. So we talked about the plasmids already, so I'm not going to spend too much time here, but let's just take a look at the visual. So when we look at the bacterial cell, remember you have two types of DNA. So you have the chromosomal DNA, which is the really long strand that's loose in the cytoplasm, but then you have these teeny tiny little plasmids, which from a base pair point of view are actually quite small. So if we look at the circular chromosomal DNA, we're talking 4 million base pairs. If we look at the little plasmids, we're talking only several thousand base pairs each. So because they're much smaller, they're much easier to work with, okay? And they actually are independent of that circular DNA. So they can replicate and do their thing, and they're not under any influence of the circular chromosomal DNA. And sometimes, if they're small enough, they can even pass through the cell membrane and like go away. We're not really sure why that happens, but it can happen. So in any case, remember when we do the recombinant DNA thing, right? These little plasmids are what are used. So the restriction enzymes are added to the little plasmid to cut out a section, which is going to be complementary to the one that was cut out of the original donor chromosome, right? So that's why they're used, because they're smaller. And then when they're reintroduced back into the bacterial cell, they're going to happily produce insulin or growth hormone or whatever it is that we desired. So this is just another graphic showing the same thing that we kind of talked about already. So here's, but this one doesn't show our chromosomal DNA. It just shows our, like from our original donor strand, it just shows our plasmid. So remember that a restriction enzyme then is added to the circular plasmid, and we're going to add the same restriction enzyme that we added to our donor DNA. And so we end up with our sticky ends here, in this case, instead of blunt ends. And then over here, it just kind of shows us this is what was cut out of our donor or our chromosomal DNA to begin with. And notice how you have this sticky NG on the bottom here, this sticky NG on the top here. So they are going to bond with one another, right? Because those base pairs are going to match up. And then after the donor genetic material is added to the plasmid, then ligase is added to glue everything and seal everything up together nicely. And then of course the plasmid's put back into the bacterial cell with that new gene so that it can make whatever protein product is desirable. Okay, so that essentially is what genetic recombinant DNA is all about. So the last thing that we're going to talk about today is gel electrophoresis, okay? And this is a fairly easy concept actually, but essentially what happens with gel electrophoresis is this is what's used, you know, when you watch TV shows, when you watch like their you know, doing genetic analysis or DNA analysis to solve a crime or to compare DNA of individuals or whatever the case is. So essentially what happens is you have a power source, so electricity is involved here for sure, and then you have a gel medium, okay, a gel medium in the apparatus, and then basically the power source is going to add electricity to that gel medium. Okay, so then what happens is in these little test tubes at the top here, you have DNA fragments of different sizes. And then the DNA is added to the electrophoresis plate. As the DNA is added to the plate, what ends up happening is the molecules are separated. So those little DNA fragments are separated based on their attraction to the negative versus the positive end of the electric field. So you can see that as the process goes on, the pieces of DNA fragments migrate, okay? And then in the very end, you have fragments that are distributed across this completed gel plate, and then that can be compared to other DNA samples to see how closely related the organisms or the humans or whatever all to one another. So the longer fragments stay towards the top, and the shorter fragments actually travel further down the plate. And then in class, we'll do some practice in terms of, okay, now that we kind of sort of know what this is, how does that impact, us in a test situation? What kinds of questions would we look at? And we'll do some practice with that.